Hey, what is up? It is Tuesday, March 29th, 2022. It is just about the end of March. 2 more days to go, and it's Metabolism March. I think we can end with a bang. I'm ready to talk almost. Yeah, I'm pretty ready to talk about gluconeogenesis, and that's why I have this up. But today's going to be a pretty short video because the process is actually quite involved, as in it requires understanding of several different processes, including the reversal of glycolysis and the involvement of the Krebs cycle in the reversal of that glycolysis. And it also involves the products of fatty acid decomposition and triglycerol decomposition, triglyceride decomposition. Um, we we can just talk about it briefly. Tomorrow I'm going to get into it a lot more, but for now I'm just going to go over why we need it, what's the purpose, and where does it occur, etc. And roughly, without going into the super crazy details, the details I will save till tomorrow. But so normally your cells, they need glucose to function, right? All of your cells need glucose to function. All of your cells take in glucose, and they send the glucose through the process of glycolysis, which I've detailed in previous lectures, and which you probably know about from high school biology or college biology. So glycolysis breaks down glucose into pyruvate ultimately, and then pyruvate goes along its different pathways depending on whether there's oxygen present or whether there isn't oxygen, aerobic versus anaerobic conditions. Essentially, glucose is, there's a limit of how much glucose can be stored within your blood, and there's a limit of how much glucose can be obtained from sources in your body. But let's just talk about what happens. So you can store about, or you need about, on average, just to survive, 160 grams of glucose a day. Okay, that's the average non-athletic person. Um, when you when you deplete the supply, essentially you don't have very long before you croak. Unfortunately, it's to say, but you don't get you take this glucose in, and eventually you take this in from your foods, and the glucose is stored as glycogen in the liver up to a certain amount, and it's floating in your blood up to a certain amount, and it's taken in. But essentially, there's only a limit to how much that can be done in this case, and then eventually, when you stop eating or when you don't consume enough food and your glucose is being depleted, it starts being depleted from the liver until it's gone from the liver. And at that point, when your glycogen is gone, then you're going to have to. That's actually essentially when you start when you exercise, you start thinking about, oh, where does my energy come from? It starts coming from the carbohydrates you've consumed, the glucose that's floating in your blood, and the glycogen in your liver. And remember, just going back, glycogen is glucose polymer. That's basically the way that glucose is stored in your body for longer term use. But once that's gone, then what do you do? Then you have to start, where do you get glucose from? Well, let's say you can't get the food, and it takes some time to digest that food to turn it into glucose and whatnot. What you need to start breaking down fatty acids. You need to start obtaining glucose from other sources. So, how do these sources come by? Where does this happen? How do you get these additional sources? Yeah, and the essentially, what is the process of this occurring? So, um, uh, you basically, what happens in gluconeogenesis is that you, in the liver cells and kidney cells, and into a small in a small part in the intestinal cells, this reversal of glycolysis occurs. So when I say reversal of glycolysis, I'm not talking about glycolysis reversed by the enzymes of glycolysis itself. Glycolysis is an exergonic process, which means it produces energy, and it releases energy, and it releases a lot of energy, especially in the so-called irreversible steps of glycolysis. The step 1, 3, and 10 are the steps of glycolysis that reduce the most and produce the most energy and heat your system up the most. And step 3 especially, which is the committed step involving phosphofructose kinase, does the most of that. So we can't just simply recreate glucose from pyruvate, because pyruvate's the end product of glycolysis. We can't just put the pyruvate back together using the same enzymes, because the process, those 3 steps of glycolysis are not in equilibrium. Okay, so we need to actually involve enzymes that are within the mitochondria to do this, to kind of recreate the products of intermediate products of glycolysis before they've gone down into the irreversible steps. So, for example, the step 10 of glycolysis is the conversion of phosphoenol pyruvate into pyruvate. We need to use enzymes in the mitochondria to recreate phosphoenol pyruvate, PEP. Okay, we can't recreate PEP by using phosphofructose kinase in reverse. It's pyruvate kinase in reverse, which is the enzyme which essentially produces the pyruvate. Okay, so, but again, going into that, I'll go deeper into tomorrow. Let's talk about what are the components. So essentially, we need to form glucose. Where does gluconeogenesis occur? Again, it occurs in the liver, in the hepatocytes, right? If I go to, where does gluconeogenesis occur? See if we can have a breakdown. It occurs mostly, yep, here you go. You have the liver here, and you have a number of enzymes involved. You also need the source of the products or source of the inputs for gluconeogenesis, which includes glycerol. Okay, so you need glycerol. We'll go into exactly how that's needed, and that's done through the fatty acid breakdown. We don't use fatty acids. Fatty acids can't be used to create glucose. We just need the glycerol, and we need amino acids involved, right? Glucogenic amino acids, and we need for pyruvate. We also need the pyruvate inputs, which come from lactate, right? So muscle cells produce lactate, and this lactate, via lactate dehydrogenase, is converted into pyruvate, which through the use of several other all these other inputs, the amino acids, which can get converted to alpha keto acids, right? And through glycerol, which gets converted to DHAP. All these 3 things combined together, pyruvate, DHAP, alpha keto acids, they form glucose, right? Okay, so that's gluconeogenesis, and it occurs by the action of the fat tissues and the muscle tissues, which provide input. Okay, so that's a quick introduction to that, and just also I want to mention why we can't just simply do, or emphasize why we can't simply just eat food, because again, your liver can only store so much glycogen, your blood can only have so much glucose in it, so you need at some point, when you don't have a food source readily available and digested, to produce this glucose, right? It seems interesting. Why couldn't your liver just store a lot more glucose? Then your liver would get huge, I guess. I don't know the exact reason, but there's probably other reasons, including the fact that glucose is actually toxic. I don't know if glycogen is toxic, but glucose in large amounts and long-lived quantities in your body has the toxicity. So yeah, we need to use it, because it's the energetic source component. Okay, so that's it for today. I'll get more into gluconeogenesis tomorrow. Thanks for watching my video, and I'll see you around.